Welcome to all for this particular session of Class 12 Biology, Chapter 9. It's plant breeding. We would like to understand a few aspects of plant breeding. You already learned some concepts, some ideas, some information about animal breeding, and now we will try to know what is plant breeding, how plant breeding is helping the Indian economy, how plant breeding is helping the Indian population, and what are the methods followed by farmers, how scientists are helping the Indian farming industry, especially plant industry, how we achieved the green revolution are all major aspects of Indian agricultural activities, and that is taken care by a particular branch of science called plant breeding. So in this session, we will learn a few basics of plant breeding. Okay, we shall now try to differentiate between two very similar words. One is called reproduction, the other is called breeding. Reproduction is a natural way in which plants produce new plants by either by sexual methods, that is involving the pollen grains and the ova, or by vegetative methods where some part of the plant body can become a new plant. That is the most natural way of reproduction. And in this thing, normally human beings do not interfere with the yield. Yield is very natural. On the other hand, breeding also indicates reproduction, but in this case, the scientist, the farmer, or agriculturist, or researcher would intervene and they will select pure lines. If I give you an example, supposing you think of a papaya plant, let's say, one papaya plant is very tall, maybe about 8 feet or 10 feet tall. It produces very sweet fruits. Let's say there is another variety of a papaya that is short, maybe about 4 feet or 5 feet. The fruits from that will not be that sweet and tasty. So now the farmers or the researcher would like to mate these two. That means selectively selected plants are mated sexually or by any other method, we'll be knowing that, and they produce a good variety of a plant, and that is called a breed. So breed refers to crossing pure lines and then allowing them to reproduce several generations, and the progeny is selected, and that progeny will be desired qualities and also high yielding, and that is, we do it, scientists will do it by using the most modern technological advances of the plant science. We shall look into that, what it is. So in this particular PPT, I would request you to concentrate saying that difference between reproduction and breeding. Okay, we will ask one more question. What is the aim of breeding? Why scientists, researchers breed the plants? Why not they allow it to happen naturally? For that, the answer would be the objectives: to research, develop high yielding and disease resistant varieties by carefully selecting progenies. That is the major objective of the breeding. It should be desirable qualities and they should be disease resistant. And then for this purpose, we have to use molecular technology, genetics, genetical methods, tissue culture, and other advanced methods have to be employed to produce the desired type of a progeny that would benefit the human beings or humankind. And that is the objective of breeding. breeding experiments successfully carried out in India has produced a large quantity of very good wheat, rice, sugarcane, millets, maize, green peas, groundnut, and all these grains have been produced by means of selective breeding. Similarly, oil producing seeds like sunflower, coconut, groundnut, gingerly are again subjected to breeding, and today we got extremely good varieties of yield and also high volume of materials are produced by this. Similarly, fiber yielding plants like cotton, jute, coconut are also subjected to breeding experiments, and we got varieties of plants now available to farmers. Nutritious fruits like dates, mangoes, banana, guava, grapes, oranges are all now genetically modified by means of breeding experiments. And today, a guava fruit will be weighing almost about half a kilo, and it will be seedless. So we have seedless dates, seedless mangoes. Sorry, mangoes are not seedless yet. Maybe they will come out with those. Grapes are seedless. So grapes in grape industry is growing very well, and large number of wineries have come up, and medicinal plants, fungi, bacteria, microbes are all subjected to genetic experiments, and they produce in large quantities. Many more plant types are created by advanced plant technology by genetic and mutation type of breeding. We shall examine what they are. But one thing we must remember, in India, green revolution happened because of the breeding experiments, and we have to thank and grateful to Dr. Swami who introduced these breeding methods in Indian soil. Having understood what is breeding, we shall try to find out how many types of breeding we can find out or are in practice today in India. One is called back crossing. In this, a plant with the desired traits is crossed with a plant that does not have desired traits but has several other traits. That means, as I told you earlier, maybe a dwarf plant which is not yielding anything, maybe a very long plant that is yielding very well. So they will produce a new plant, new variety, which is neither short nor very tall, but will have the capacity to yield extremely desired qualities. That is called back crossing. And we have inbreeding. That means they allow heterozygous plants to inbreed within themselves, and many generations will pass, and we ultimately get a progeny for pure line. So we may expect very sweet oranges, maybe very sweet mangoes, mangoes containing more fleshy material. All this type of the breeding is possible. That has been achieved especially in the grains like wheat and rice. You have rice variety which is very dwarf and also very short duration. Earlier days, people used to wait for 8 months or 9 months or 6 months to get a yield. But today, within a month, a farmer can get the matured rice for the market. That is the type of thing happens. Hybrid breeding is there. In this, two different breeds are crossed to produce the offspring that is more productive than the parents. So that is another type of breeding. And mutation breeding refers to the genetically they change the genetic or genome. They will make mutation to occur, and they change it, and they use such breeding also possible. And genetic engineering, where at the cellular level DNA is sliced, and they also recombine DNA. That means recombination DNA is possible, and also tissue culture is possible. So these are all the various types of breeding that happens. We know in this particular session of 30 minutes, I will not be able to explain much to you. We are only giving a brief understanding that plant breeding has many research availabilities, research areas, and you can think of becoming a researcher in the plant agriculture area, or in the tissue culture area, or in the breeding area. That's the whole idea why breeding is introduced in the class 12. Not just for examination. It is the information you will get it so that you can advance your studies in different fields. That's why I would like you to go through some of the websites and find out types of breeding and get interested in something so that you can pursue your studies future. Okay, now we come to a place when we knew something about breeding. We knew types of breeding. Now let us know how many steps are there in the breeding. Normally, how many steps a scientist will have to follow. The first one is the extraction and collecting and storing of germplasm. We will explain what is germplasm in the next PPT. And evaluating the selected plants for their quality and other areas. And then again, such plants are selected, plants are taken, evaluated plants are taken, they are made into cross hybridization takes place on them, and again from those again they get the progeny or the new plants, and again they are selected and they select the superior progeny. Superior progeny means ones known for its particular trait, which will be often produced many times even though it has been repeatedly used. That's called superior progeny. And then they test that in the field. After testing, they release that to the market. They market it, and it will go to the farmers, and they grow, and the yield will be quality type, quality wise, and quantity wise yield will be much more. So there are 5 steps that a researcher will have to follow to bring a new variety or a new breed. By breeding, we get new varieties. Rice has many varieties. I don't think I can tell you all those things in this particular session. Maybe next time we will try to see what they are. But remember, there are 5 steps. One is collection of germplasm, evaluation, cross hybridization, select superior progeny, and test, release, market, and grow. 5 steps, please remember that. Well, I told you in the first one, collection of germplasm. What do you really mean by germplasm? Let us try to know. See, germplasm is a living tissue which is taken from plants and that can be grown into a new plant. That means it can be a seed, or it can be another part of another plant part, maybe a piece of leaf, maybe a piece of stem, maybe a pollen grain, or just a few cells that can be turned into a whole plant. That is, the entire technology is like that. And so they collect well-known varieties of germplasm. It may be anything, as I told you, any part of the plant may be germplasm, including a single cell will become a germplasm. Germplasm contains the entire information for a species which has got its own genetic makeup, and that will be available naturally available in the source of a plant diversity. That means you can collect the genetic material either in the form of say cells or any part, which can be further analyzed for the diversity, and that's where we collect large number of things. You can see there are 3 diagrams given there. And the left one, seeds are collected. Very good yielding seeds are collected, and they are called the germplasm. In the second one, they have collected the tissues, and tissue culture is going on for the well-known species, what you call super progenies. And in the third one, small cells are collected, and they are all kept in the library. So there is a germplasm library available in the breeding centers. So farmers can go and ask what they want depending upon the type of the soil, depending upon the environmental condition, depending upon the labor they have, depending upon the mechanization they have made. They can easily get the help from the research center, Germplasm will help what they want. Now we come to the second step called evaluation. What is really evaluation means in this case? It is not evaluation of your question and answer. It is the evaluation of the pure lines that they produce from the germplasm. When germplasm is made to grow, made to become a plant, and that plant will produce the seeds, and those seeds are again tested and again tested and tested again. That's called evaluation. So they evaluate a particular progeny for the desired qualities they wanted, and then they come to the third step. They take such tested material, tested seeds or progeny. They come to cross hybridization. Two known tested, that means two known pure lines, two known super progenies are selected, and they are bred. They are made to hybridize, and they will produce the hybrid plants. And these hybrid plants are again planted, and they get the progeny from that. They study the progeny and find out which particular hybrid has the quality what we wanted, what the scientist wanted, what high yielding or what kind of volume of thing they wanted, what height, what color, what taste. All these things can be tested in the laboratory, and it is collected. Then they select superior progeny. From there, they collect the seeds again, or the germplasm again. They again make to them to grow, and they will collect the seeds again or the plant pot again, and then they will put it for the testing in the regular field with the help of the farmers, and they will release the seeds now with a known labeling. They will label what kind of, for example, IR28, for example, Indian rice. You can say I want IR48. Wheat, you can say I want Sona. So like that you can select the things, it go to market, and then it grows, the yield will come. And that is the 5 steps now used in the breeding experiments. Okay, you please remember these 5 steps, don't forget it. I have just shown 3 images in which we say that selective breeding takes place, that is the test is done first, and they get the second generation, and they get the third generation, like that. So from every stage they select the plant, and these plants are again tested, evaluated until they are very sure they get a pure line. That is these 3 diagrams are indicating those things. It's only repetition of what I told you right in the beginning. You can study these diagrams slowly. I think that would be enough to understand that breeding is a process where several times selection goes on, and finally they get the pure line. I am almost coming to the end of the chapter. What I want you to remember would be, at least try to remember difference between reproduction and breeding. You should be able to differentiate and explain what is reproduction and what is breeding. Explain the 5 steps involved in plant breeding. You need not know the entire details of what happens. At least you can remember the 5 steps which I mentioned to you in my presentation. And you should be able to at least tell why breeding is done in our country, what is the necessity of doing plant breeding. This question you may have to answer using your own words. Are there any dangers by creating new breeds? See, we do not know whether new breeds, whatever we are getting, how it is going to help or how it is going to create dangers in the human beings is not exactly known, because only since 25 years we have been going through the breeding experiments, and plants are bred and we are getting new varieties. But if you look at some of the things, it makes me to think in a different way. If you bring a papaya or a banana today, keep it outside for one day, it looks like green. The next day it becomes completely ripe, and the third day it becomes rotting. In the earlier days, it was not so. Naturally grown plantain can be kept for nearly 6 to 7 days, but it is not so today. So we really do not know what is the effect of the breeding experiments, and we should be able to know what happens on that. Think of it, imagine it, try to put your ideas and say right or wrong, try to find out. Then also try to understand the question, what is green revolution? These are very important areas you should remember for your 12th examination also. Of course, finally, I have come to giving my grateful thanks to NCRT for the content I am using from your textbook of Class 12 textbook biology. Google gave some information to me. Creative Commons, I've taken some images. YouTube also for publishing this for 12 standard. As I told you earlier, in a short brief of about 25 to 30 minutes, it may not be possible for anyone to give complete picture of plant breeding, but we can only say that plant breeding is a very useful technique in which technological advances have been used to produce new varieties which are high yielding and also quality wise. But no one knows what would happen to those breeds which have been there. As I told you earlier, only about 25 to 30 years since breeding is going on, we are not sure what would be the future of breeding experiments. We shall wait, watch, and see what happens to it. Thank you.